Okay, well, welcome to all our viewers. Today's event is going to be released online, and it's in lieu of a series of events that we now can't have because of the COVID-19. So we're hoping to give you some indication of what's involved in collecting, identifying and recording various invertebrates that you might find in our wonderful countryside, or indeed in your garden or on your windowsill at home. I'm Mike Clapham. I'm chair of the Northwest branch of the Royal Society of Biology and a retired lecturer, and our intrepid bug collectors beside me here are Alan and Hilary Bedford, also both retired lecturers, and they've been providing us with various insects and the like to show details of how we might identify them, record them, and encourage you to do the same, because recording of some of these groups is very dire. There isn't much done at all for some groups of invertebrates, so you could actually be contributing to scientific advancement, particularly here in the northwest of England.
Welcome to the first online workshop from the Northwest branch of the Royal Society of Biology. We're looking at collecting and identifying invertebrates. This was part of a set of workshops we planned to run, but unfortunately COVID-19 had other ideas. We're based today at the Wyre Estuary Country Park, and we're very grateful to the staff here and to Wyre Council for allowing us to use their facilities and for giving us their full support. And just for interest, we're actually based here at the Ecology Centre, and we're using the grounds immediately behind the building, which are very rich in wildlife. So why are we looking at invertebrates? This is a slide which was produced by Opal, who ran a citizen science project on a similar sort of scheme, looking at invertebrates in hedgerows, towns and so on. And if you look at the figures on here, you'll see worldwide we've got 1.3 million species of invertebrates compared to 60,000 species of vertebrates. So an enormous difference. Now, as far as most people are concerned, they're quite happy with birds, mammals and the like. They know a few invertebrates, the more showy species like the dragonflies, butterflies and so on, but there's a myriad of species which are not appreciated or actively disliked. So why do we need records? So why do we collect invertebrates and actually identify them? If you go to the NBN, the National Biodiversity Network, you'll find that they have 200 million animal records. Of those, 164 million are birds, and within that they've got 8 million records of robin. There's a very, very distinct bias towards birds and on from that other vertebrates. Clearly there's an awful lot of invertebrates not being recorded. The group I'm particularly interested in, which are the flies, which have 12 times as many species as birds, have only about 900,000 records, and they again are biased to certain popular groups like the hoverflies. There is also this strange situation in the northwest where we seem to be under-recording. Now this is the yellow dungfly, a very attractive beast, obviously is attracted to dung, but also is a very important pollinator. And if you'll excuse the pun, it's as common as muck. If you actually look at its records, although there's quite a few nationally, up in the northwest here we've got a limited number of records. The Wyre area does better than probably the area where I live down in Preston, but there still seems to be a big void area for recording in the northwest. And if you look at this other chap here, which is the orange-bearded blowfly, lovely name, Caliphora vomitoria. This is a key player in forensic entomology. It's one of the flies which colonises corpses of any sort very, very quickly after death. And again, you can see very immediately there's this big sort of void area in the north. And if we come into the Wyre and Preston, very, very few records indeed. Now, there are issues often with common species that people don't bother to record them because they're common, but more often than not it's a fact that there are very few people actually looking at flies, identifying them and have the confidence to actually go to species and submit them to their county recorder. Okay, so we're going to be talking about collecting invertebrates and identifying them, but what do we actually mean by identification? We have to look at, first of all, at the hierarchy. So if we look at this individual that we've got here, I think most of us would be very happy to say that was an animal and an arthropod, something with a hard exoskeleton with jointed legs. Most of us, I think, would also be quite happy to say that was an insect. Now, we're identifying this, but we're at a fairly unrefined level, and that really doesn't tell us a great deal. If we start to think a little bit more about which insect it is, well, it's one of the true flies, one of the groups I like. And if you go beyond that, we get to the hoverflies, the Syrphidae. So it's becoming much more useful information, but if we can actually get to species level, so we've got Volucella and we've got zone area, that's its proper international scientific name, then we know precisely what it is, and that gives us a lot of information because we can now begin to actually look at what's known about this particular fly locally and actually nationally. And here's one I actually photographed in Penryn in 2017. The interesting thing about this was this particular fly used to come across to Britain occasionally up until about the 1940s, and then it became established in London and gradually moved down the coast into Cornwall. Penryn's just outside Falmouth, but also this was actually photographed on the Sefton coast in 2018, because not only did it spread west, but it also spread northwards, and the thinking is that this was almost certainly linked to climate change. So we're beginning to get an awful lot of information about this particular fly and its importance. Much more generally, if we take a place like the park here, the country park here, they want to know what's actually living here so that they can actually look at whether they've got the species they should have, whether they've got any species which are unusual. And if you monitor long term, then you can actually see whether or not the habitat, the local habitat, is actually changing either for the better or for the worse. So what we're going to do now is we're going to go and actually look at some of these invertebrates, or how we actually collect them, because although there's loads of them actually here, they don't go out of their way to make themselves apparent. Right, so we want to get some invertebrates that we can actually identify. Now, there are loads and loads of species here, but an awful lot of them don't go out of their way to make themselves visible. How do we actually find them? Well, the easiest way to start with is direct searching. So if we actually have a look at some of the plants here, we can have a look under the leaves themselves, and we may actually find invertebrates. We'd be looking for particularly the slow-moving or even completely sedentary invertebrates. We may see others, but they tend to be a little bit more active and will tend to fly away. And so if we just have a hunt around in the vegetation, we may find something. We're on camera, so there's very little chance, but there we go. I don't know whether you can actually see down here, we've actually got some flies on the leaf, but we'll come back to those in just a moment, because I suspect they're not going to allow us to actually catch them. But if we do find something, then all we need to do is actually put it into a very, very basic pot. Now, in order to help you, there are various bits of equipment which you can use, and most of these are actually really cheap and easily accessible. Probably the most important piece of equipment is the pooter. And the pooter is very straightforward. If I just show you with this large one here, you've got a tube which you suck on, and you've got a tube at the end where hopefully your invertebrate will go in. Okay, and always you should have at the end of the tube where you suck a piece of gauze which stops you actually swallowing the flies themselves. Now, on a relatively cold morning like today, some of these more active beasts might actually be just a bit docile. So I'm going to go and try and advance on this one here. Give it a go. And in it goes. Okay, so I've got my fly actually in the tube, and it actually has been traumatized. It'll come round in just a moment, but the very act of being caught has actually made it relatively immobile. Now, that's my favourite pooter, but you can buy these very, very easily online, made of plastic, or you can have the older ones actually made of glass, but obviously glass is a little bit dodgy when you're working out in the field itself. Let's have a look and see if we can catch... Oh, wait a minute. Oh. And Hilary has been pooting while we've been talking, and we've got a nice range of invertebrates actually in there, and one of them's actually trying to escape. Now, when we come to identify these, we've got two basic approaches, and I will show you. Now, you'll notice that these flies are really quite active. We can make them less active by a number of ways. One is to take the pot home and put them in the fridge for a while, and they will slow right down. A lot of wildlife photographers do that when they're actually photographing insects. We can, we'll try it with this one here, give them a dose of carbon dioxide. So bear with me a moment. Okay, and you can see that has knocked them out immediately, and that means I can get a closer look at them with some like a hand lens. I'll just take them out and put them on my hand, and they will probably come round while I'm actually talking. Now, in fact, you can see one or two of them beginning to move their legs. It's a very short-lived way of anaesthetising them, and it does them no harm at all. All I've used there is one of these tools you can get from cycle shops for blowing up bicycle tyres. So it's a carbon dioxide cylinder and a nozzle, and you can see they're beginning to, see that wasp there is beginning to twitch a little bit. All of them, I promise you, will come round fairly shortly. Come on then, in fact we'll take them with us. Now, many of the invertebrates you're wanting to actually collect are things that live in the soil, or they live in leaf litter. They're invertebrates which are poorly adapted to life in the terrestrial environment, and they dry out very, very quickly. So if we put something down like a piece of roofing felt, very similar to what people use actually when they're doing reptile surveys, things that, like the dark and the moist, will tend to move in, and they move in very, very quickly indeed. So if I ask Hilary very nicely, and she'll lift it up, and we'll just have a quick look at what's under here, and you will see moving around. They come in. Okay, we've got a couple of woodlice there. Obviously you've got to be incredibly quick, because the minute that you start exposing them to the light, obviously they move away, and of course you get these gorgeous cute slugs. Slugs and snails will move in under a piece of roofing felt very, very quickly. Now that bit of roofing felt, we've got another lovely slug there as well, and we've got woodlice. Yeah, we've got several species of snail there. This has only been out for a couple of days, so if we have covering which has been there for longer, we're likely to get more. And it just so happens, if we go around the corner here, we've got some building sacks which have been there for several weeks. And we've got a nice earthworm just under here, another one over there, lots of woodlice, a centipede over there, and Hilary has got a lovely ground beetle. She caught it, there we are. And we've also got a millipede in this pot here, so let's just hold that up to the camera. I mean, clearly he doesn't want to hang around for very long. Hilary did very well to actually collect him. But these are the sort of invertebrates you're going to get hiding underneath things in rotten wood and the like. A lot of potential there, but obviously you've got to be fairly quick with some of them. Now, for those more active invertebrates, particularly the insects, then netting is going to be the most useful way of actually catching them. They are not going to hang around while we try and poot them, or whilst we try and grab them in a tube itself. And when you're netting, what you're doing is you are disturbing them on whatever their substrate is, and you're trying to catch them as they try and fly away. The intention is never to actually mow the lawn with your net or get vast amounts of vegetation in your net, because that just gets in the way anyway, and it's extremely damaging. Now, as far as nets are concerned, they come in all sorts of shapes and sizes. This is a classic butterfly net with a long handle, very useful for reaching things which are a long way off. and in fact for any net, being able to add a handle later on is really quite useful. We've got here these ones which have a very rigid frame. These are sweep nets, which are very good for doing numerical surveys, and we'll demonstrate those in just a moment. And this is a really useful general purpose net that you can use for all sorts of sampling. This particular one, it comes as a folded frame, so it's extremely useful for packing away in a rucksack, and I'm not going to assemble it completely, but you've got the rim, you've got the netting, and there you go. Now we've got two colours. We've got a black one here. Black one is very good for butterflies and dragonflies. They're nice and big and you can actually see them in the net, and they tend not to see the black netting itself coming. White nets are used for most other insects in particular, because when you've actually caught your insects, you can see what's inside. And particularly with smaller individuals, it's quite useful to know what you've got before you actually start actually looking. Okay, so what I'm going to do is I'm going to show you how, first of all, we use a sweep net. Now the conditions here are not actually ideal, but we've got a nice relatively open area here, which is very good for really swinging the sweep net itself. It's got a bit of bramble here and there, so we'll have to try and watch out for that. But the idea of the sweep net is you're sweeping through the vegetation, you're just actually skimming it. Every time you've swept, you come straight back into another sweep, so the insects don't get a chance to actually escape. And then when we get to the end, you put a little flick on like that, so whatever you've caught is actually trapped in the back of the net itself. Now we'll have a look and see if we've actually caught anything at all. I said you're not trying to mow the lawn. Well, I seem to have got a certain amount of vegetation here, but let's just open it up and let's have a look. And immediately you can see we've got flies, and they're all starting to actually escape here. But there's a fair number there, probably better than I expected, and there'll be a few more bits and pieces probably associated with the vegetation itself. And we've got something green which is crawling up there, probably a bug, but a nice fly here. Lots of flies is what you actually tend to actually get. Now of course, on a spider, rather nice spider. Now of course they've all escaped, so that's a bit daft, isn't it? So there are techniques for actually getting the invertebrates out. So what I'm going to do is I'm going to do another sweep, and then we'll go back over to the meadow over there, and I'll show you how I normally get them out of the bag itself. Okay, so we've got our invertebrates in our net. How do we actually get them out? And there are various ways of doing that, but the best way is to use our friendly pooter. You can open the net and try and actually catch them as they come out, but the best way is to go in and get them. So, go into the net, hold the net up towards the light, and they will, in theory, try to go up towards it. Okay, I've just collected a small fraction of what's actually in there. In fact, I did better on that second sweep than we did on the first one. As you can see them coming up. Might better catch one or two. Give it a go. No. Come on. There's one. Got it. Now the nice thing about putting your head in the net is you can select what you actually catch. So you can avoid things. If you've got lots of the same species, then you only collect one or two, you can let the rest go. Just watch out in case you caught the odd bee or two, or a very angry wasp. They don't tend to like being in the net. Best to let them go first of all. And if we look, oh, drop that on the ground. So here we are. Those are some of the invertebrates which I took out of the net just now. Now in longer vegetation you can't get so much of a sweep, so you've got a short, sharp action. This will dislodge a lot of invertebrates, and hopefully we've got a few of them in the net there. You can see the net is somewhat wet. Oddly it's been raining up here in the northwest, which is very strange, but never mind. Okay, now how do we actually get those invertebrates out? Because I can't get my head in there, I'm far too big-headed for that. So an alternative method is you invert the net into a plastic bag. Okay, so as soon as Hillary's ready, and you simply push it, so like pushing out the finger on a glove, and shake them all off. Okay, and then pull the net out, and away we go. And we didn't do desperately well as a catch there. Sorry, you've got one or two flies actually in there. In fact, actually we begin to look, there's a spittle bug there. We've got one or two caterpillars, and we've got some of the smaller flies. On a sunny day, we'd get shed loads of stuff actually from an area like this. I'm afraid the weather is not on our side. Now, another way of actually collecting invertebrates in the field is to use what's called a beating tray, and Hillary's holding here what's considered an old-fashioned beating tray now, but very, very effective. Something you hold under the branches. You can get ones like this, which are much more recently produced commercial ones, and you'll pay an awful lot more money for them. But the principle is that you go up to your tree or your vegetation, and you give it a hard bang, and if that doesn't work, you can give it a good old shape, and you might get the odd insect coming off, but mostly we seem to have collected apples here. There doesn't seem to be an awful lot on this particular tray at all. But what you're interested in here are relatively slow moving and mobile species. Anything falling into your beating tray you would then collect with the pooter itself. Now, an obvious solution for this sort of work, rather than buying yourself a commercial beating tray, is to use a white umbrella. And particularly if you've got one, this is a golfing umbrella, but if you've got an ordinary one with a hook on, you can even hook it on the branch and then bang into it. Very, very effective. Everything, in fact, we've caught something already in here, but everything falls into the middle. They actually have trouble escaping, so it's very, very effective. And of course, on a day like this, you can also protect yourself from the rain. Now, the techniques we've listed so far, we've used so far, what are called active methods, because they involve you almost breaking into a sweat. There are other techniques, passive methods, which can be used. These are particularly useful for collecting nocturnal species, species that move around a great deal, or for things which are buried deep in vegetation like this. And if you were to crawl around on your hand and knees chasing the beast, you'd probably cause a lot of habitat damage. A very simple and easy one to use is what's called the pitfall trap, which is a plastic cup of some sort which is sunk into the ground so that the lip is level with the ground itself, and animals then just simply walk into them and fall in. Okay, so I'll just quickly demonstrate. So find yourself a convenient spot, clear down to the vegetation itself, something like a bulb planter. These old bulb planters are brilliant for this, and you simply cut a hole, and of course I found a nice hard bit, but never mind. Okay, like so, and then you insert your cup so that the lip is level with the soil surface. I find these really tough food preservation pots are very useful, because you can just put your foot on top and actually push it in if they're strong enough. If you leave the cup with a lip projecting, invertebrates simply follow the lip and walk around it. You want them actually to fall inside. And then what you want to do is put some sort of cover on. Lots of ideas for this, but old tiles or bits of slate, that's put on, supported off the ground by about a finger's depth is what I use. And the idea is then that discourages things like newts and small mammals from actually falling in as well. Now, as far as the pitfall trap is concerned, you've got various options. If you want things to remain alive, then you've got to put food in them and places for things to shelter, bits of newspaper, leaf litter, and so on. But you are going to find at some point that predators will get in, things like the ground beetles, and they will tend to eat things. So mostly what we do is we put some sort of killing agent and preservative in, so that the animals die and they are preserved and don't start to actually rot. It used to be very common to use car antifreeze, ethylene glycol. It's very, very effective. The only problem is it's very sweet tasting and it's poisonous to dogs in particular. So in an area where you, particularly like an area around here, not here, but actually in the park as a whole, where you've got lots of dog walkers, it's a non-starter. There is an alternative, which is propylene glycol, which is actually used as a food additive, and it's used in the drug industry, a close relative of ethylene glycol, and that works extremely well. So would strongly recommend that. Now, we put a couple of pitfall traps out about 3 days ago. I've used tile here. This is the core of soil which came out when I positioned the pitfall trap. I'll mention that in just a moment. But if we simply take that out, pull it out there, and we've got a range of invertebrates there, nicely preserved, that we can identify. Okay. Now, having finished, that goes back in the hole, because otherwise you end up with a permanent pitfall trap and things will get caught in there and they will be eaten by predators or simply die for no reason at all. So in they go. Okay, this is what's called a water trap, and it's very effective for flying insects, the sort of insects that might cover big distances and you'd be hard pressed to actually catch some of them. It's very, very simple. It's a plastic tray, a yellow one. I've painted it yellow on the underside. It's got water with a dab of detergent in, and flies are attracted to the yellow colour. They hit the water, go straight in, into the water. Because of the detergent they sink and they will drown. There is nothing you can do as far as this is concerned to catch things alive. There's not an option. You know this is going to kill things. So if that's not your beef, then don't do it. If you are going to use things like water traps, then they should be used as part of a survey, so that when you identify the specimens, they will then contribute to the record for a particular site. They're not a sort of thing you use just to see what's actually there. But they're very effective. They particularly catch hoverflies and really a whole range of other fly species, and we've got quite a nice little range in here, including one of my favourites. It seems a strange thing to a favourite to kill it, but there we go. These little ones here, which are what are called the dollies or doe-lycopodids, the long-legged flies. They are beautiful brassy colour, or they're metallic green, very, very attractive flies, and voracious predators at that.
Good morning, I'm Dave McGraw. I've been a naturalist for a very long time, since I was knee-high to a grasshopper, and in the last 20-30 years I've been getting into moths. And here we have my moth trap. As you can see, we've got a very powerful lamp which emits a lot of ultraviolet light, similar to what you'd see in the butcher's shop, hanging sort of near the door to make sure the flies don't come in. If they do, they get zapped, but this is much more powerful than those. And the moths are attracted to it, fly around, and then eventually tire and bounce off the sides of the perspex sheets and fall through the hole down at the bottom. Once they're in the trap, you can see egg boxes laid around, and the moths will hide in amongst those and stay there quietly for the rest of the night until the morning comes, and we can visit the trap, take the perspex sheets away and see what we've found. So to collect the moths, we have a variety of size tubes for the smaller moths, the micro moths, and then larger ones for the macro moths. And if we're lucky enough to get any hawk moths, we've got some larger pots that they will fit into. Try and keep it all together neat and tidy. And of course you need to know what you've caught, so we have 2 field guides. We've got one for the micro moths, and then we have the other one for the macro moths there. Just looking at the cover, you can see that some of the moths are far more colourful than a lot of the butterflies, and you can give me an emperor moth over a meadow brown butterfly any day of the week if you don't mind. And of course the most important thing with your catch is to have your notebook ready, waiting with a pen to record everything you find, and then log it with the appropriate authorities. I send mine to the county moth recorder, and from there it goes on to the national database, so that wherever we trap we've got the record of what's been caught and how many, etc. which is important for the conservation of the species. Moths at the moment are undergoing quite a significant decline. Something like 30 to 40% of all moths have been lost in the last 30 to 40 years. So they're like many other insects, they're declining at an alarming rate, and they're important creatures because they're pollinators, just like bees and butterflies that everyone knows and loves, and hoverflies too. At night it's moths that do a lot of the pollinating, and of course the caterpillars are at the bottom of the food chain for birds like blue tits, great tits feeding their young, and the adults are eaten by bats at night too. So they're incredibly important creatures, so we need to know how many, what species, and how they're doing in different parts of the country, which is why there's loads of people out all over the British Isles trapping moths every night of the year really. Even in the winter there's some active, but most obviously are found during the summer months when it's warmer. Yeah, so if we get on to open the trap, I can see that before we lift the lid we've got a common footman down at the bottom there, so it's fairly cool, so hopefully it won't fly off when we bring him out. Put that across there to just lift this one gently out of the way, turn him over. No one home on that one, so lift this one out, and there's our common foot. Looks like a pumpkin seed. Just put the paintbrush over. If it flies off it doesn't really matter because I've already recorded this one in my notebook. If it goes on the thing, it's got that gorgeous little orange stripe down the side and the little lead coloured wing cases, making it very obviously common footman. One we see quite commonly around gardens and sort of woodland edges. Turn this one over. You'll notice there's holes in the bottom of here, and that's if there's a moth stuck in the bottom. I can poke them out with the paintbrush. Have a pot ready over the end, something like that. So there's one caught in the bottom. You can put the pot over and then poke through and drop him into the pot and bid him up. No one home in that one. It wasn't the best of nights last night for moth trapping. It was mild, but it was very windy, and it was quite heavy rain for most of the night. So I'll move these across, lift the other one, lift this one out of the way. Let's see if we can find this fella. He's still down at the bottom. So no one in that one. Very gently. Oh, who have we got here? Oh, this one's called the Flame. It's looking a bit worn, a bit worse for wear. Very obviously a little tightly furled, wider at the front, at the back, little white face. So that's moth number 3. Oh, here we've got very common moth. This one's heart and dart. You can see the dart mark. It's a nice fresh individual. That's the heart at the bottom, and then the dart marks, the dark stripe is the heart. And they have a little set of eyebrows too. It's quite one of the most common moths on the wing at this time of year. That's a nice fresh individual, very nicely marked. And that's the end of the moths.
So we've looked at techniques for actually collecting our invertebrates. Now we have the task of actually identifying them. Now, on the market there are some very useful guides, but do be a little bit careful. They're very useful, particularly when you're looking at something and you haven't got a clue what it is. You can flick through the photographs and get some sort of idea. But don't imagine that these have all of the insects in these particular cases which occur in Britain. This book, for instance, has 2,700 photographs. There are, in fact, 24,000 insects in Britain, so there's no way that can actually cover everything. So don't use the species names, but use these as a guide to get you to roughly what you're looking at. And with the Collins guide, it does say at the top, common species. What we're going to use, or what you should start with, is this identification key, which was produced by Opal as part of their citizen science project we mentioned earlier on, and you can download this from the website actually listed here. And it's a very simple guide, but it's ideal for people who are not that familiar with the various groups of invertebrates. And if you look down the bottom here, you basically start, and it's all to do with legs. No legs, 6 legs, 8 legs, and more. And when you look at the sort of invertebrates we've been seeing this morning, things like snails and centipedes and millipedes, clearly there are big differences in terms of the number of legs. Once you've actually counted the number of legs, or lack of legs, depending on what you're looking at, then you go on to subsequent sheets, and this has more information. So here we are, we're looking at the section of the key on mollusks and annelids, the snails and worms. These obviously have no legs at all, but do be a little bit careful. We all know earthworms, or we think we do. The key thing about earthworms is, apart from having no legs, they have segmented bodies. There are things out there which may confuse you. For instance, the New Zealand flatworm, which is quite a chunky beast. It has no segments and it eats earthworms. So you may well come across those. They're certainly present in the northwest of England. Now, if we then move on to myriapods and crustaceans, these are the animals which have a lot more legs than the classic insects. So woodlice have 7 pairs of legs, centipedes do not have 100 pairs of legs, they have at least 15 pairs of legs, and millipedes have probably more. So we've got under the microscope here a typical millipede and centipede. On the right we've got the millipede, and you can see quite clearly there that it's got 2 pairs of legs per segment. The legs look very crowded on the body, and millipedes characteristically are quite chunky and they're almost circular in cross section, just a bit flattened on the belly. Centipedes, on the other hand, that's the other specimen we're looking at, that's got far fewer legs, 1 pair of leg per segment. They're typically flattish animals and they have piercing mouth parts, quite chunky mouth parts. Okay, so under the microscope here we've got a woodlouse seen from the side. It's got 7 pairs of legs. You can see at least 5 pairs of them quite easily there actually in focus, and it's also got 2 pairs of antennae, which is quite useful because this is a crustacean and you don't get that many antennae actually in the insects. Now this animal here looks superficially a bit like a woodlouse. It's fairly flattened. Woodlice are always very flattened, and this one, like many woodlice, is capable of actually curling itself up into a ball. You can see immediately it's got more than 7 pairs of legs, and if you look carefully, particularly towards the back end, it's got 2 pairs of legs per segment. So this is actually a pill millipede, and it's the sort of thing which can catch you out when you're first starting to look at these sort of groups. Out of this group of 3, this is the final one we're looking at, and again, superficially this looks a little bit like a centipede. It's a flattened animal, but we've got the 2 pairs of legs again, so we know this is a millipede, so it's a flat millipede. All of these things are there, and they're going to catch you out when you first start to look at groups that you're not familiar with. So after you've dealt with the true flies, you end up with a whole raft of insects which actually have 2 pairs of wings, and it's the wings which actually cause people quite a lot of trouble. If we look at animals like the beetles, for instance, initially people think that they don't have any wings at all. These are the front wings, 4 wings, and they're hardened to form what we call elytra, and they meet down the middle perfectly, right the way down to the end of the abdomen. What they've done with this key is they've separated out beetles on the basis that the elytra, these 4 wings, meet the thorax to make a sort of T. What's not obvious at all to anybody is that underneath here is a second pair of wings, which are the ones which they actually use when they're flying. If you ever see beetles flying, they have a very ungainly way of doing it because they seem to be back end heavy and they sag and dangle their legs. Under the microscope here we've got the back end of a beetle, and here you can actually see the elytra are actually parting. So these are the 4 wings, they're parting and just peeking out between them are membranous bits of the hind wings themselves. Now, under the microscope here, we've got another beetle. This is a rove beetle, or staphylinid. These cause trouble in their own right because the elytra are really quite short and square and only cover part of the abdomen itself. But this one's really nice because it's got its hind wings completely showing. So these would normally be tucked up, folded very neatly underneath the elytra themselves, but we can actually see it's got obvious second pair of wings. Another group which causes some confusion, another important group, are the true bugs. And you'll see, if you look at what they've done, is they identify these or separate them from the beetles by having either the wings meeting as an X or meeting down the middle but forming this Y shape because of this projection of scutellum coming down the middle here. Most of the wings are hard, though, similar to the beetles themselves. So this XY arrangement is quite a useful way of actually separating them. On the screen under the microscope here, we've got a shield bug, and you can see the hind wings. The head is down to the bottom right of the screen and the tail is up to the top left. So they've got over the abdomen some quite hard sections of wing, and then those are membranous at the tail end and actually cross, forming this X shape that they mention on the key itself. Now we've got another bug under the microscope now, and this one's lying on its back, and this shows actually what is the real characteristic of bugs, which is this long piercing mouthpart called the rostrum. And bugs typically are either plant sap suckers, or they are predators and they suck the blood out of often other insects. So in many ways that would be a much more useful characteristic for separating out the true bugs. So this is a really useful introductory key, but it has its limitations because there are so many different variants which actually have to be covered. You should be looking to go further than this, and would strongly recommend Tilling's FSC, a key to major groups of British terrestrial invertebrates. This is a much more sophisticated key, and it'll allow you to go much further in terms of your identification. So if we have a look at that key, don't bother about reading this particular screen. This is page 1, key A within the Tilling's key. And it's what we call a dichotomous key. So you have pairs of questions, and on the basis of your answer, you go to one or two routes through the key itself. Just looking at the introductory key, if you go right to the beginning, it asks you about whether you've got legs or not. Very similar to the opal key that we mentioned before. Then it asks you how many pairs of legs you've got. 3 pairs of legs, obviously, for the insects, or more for the other groups like the myriapods, isopods and so on. If we've got 3 pairs of jointed legs, then it starts to ask about wings, much more specifically, and when you get to wings, then it asks you how many pairs. So we're back to this idea of how many wings have we got, and we do have to get to grips with the groups, which actually can be slightly misleading. Now if we're thinking about a true fly, like our hoverfly we mentioned earlier, then that's only got 1 pair of wings, and that will then take you to a particular section of the key dealing with all insects which just have 1 pair of wings. And sure enough, we've got our true flies, the Diptera, actually come out at 2 points here, but we've also got this group here, the Strepsiptera, which only have 1 pair of wings, and some mayflies, and 1 or 2 other groups as well. And if within the key you follow the route where it says no wings, Diptera pop up again, because some Diptera, some true flies, are parasitic, like ones that live on bats, and they've got no wings because they don't need them. Why have wings when the bats are going to fly you around anyway? So this is Volucella zonaria, the hoverfly that we mentioned in the introduction, which is also called the hornet hoverfly because it looks like a hornet. And it's one of these mimics. So do be aware of mimics. They pop up all over the place. Here's another fly which is a mimic, which is not a wasp. The hornet hoverfly wasn't a wasp. This isn't a wasp. This is what's called a conopid. And I put this in mainly because I think they're quite entertaining. Apart from being black and yellow, so they have these warning colours that the bees and wasps actually use, this particular group actually lay their eggs inside of bees. They intercept them in flight, and the female uses this structure called theca, commonly called the can opener, to actually prise open a hole in the cuticle of the bee and insert an egg. Pretty nifty, I think. The hoverflies, quite a few of them actually are mimics, and whatever you may think, these are hoverflies and these are bees. This is a hoverfly which mimics that particular bee, and these two are hoverflies which mimic that particular bee. So please be aware, mimicry is very, very common, and it can mislead you. Do take the time just to check backwards and forwards in the key, otherwise you'll end up going down the wrong route and wasting a lot of time. Now, there are various things you can do to take your identification forward. Some of you may be familiar with iNaturalist. This is an app that you can have on your mobile phone. You can submit a photo and it will give you, if it can, an almost immediate identification. It's a bit of a cheat. It's useful for confirmation, but if you don't get to grips with keys, then you won't learn a great deal about your various invertebrates. But this can be very useful for confirming ID or helping you with things that you're not familiar with. iSpot itself has a whole string of online keys where instead of you going through a paper key, it asks you a series of questions and it will lead to an identification. You may well find that you're interested in a specific group. I bring up this one because it's a fairly, people might think, a fairly obscure set of invertebrates, the myriapods, millipedes and centipedes and the isopods, but there's a very active support group and they will direct you to where to get detailed information on identification. Often they run recording schemes so you can submit records and get feedback. When we were thinking about this course, I thought what we must do is put together a list of all the online links which are available to take you to further information. And then I came across the Bexley Wildlife website, and they've done that already for me. It's got a fantastic list of links directly to websites with identification keys, helpful information. I'm not going to waste my time showing it you. Do use it. It's extremely good. And lastly, if you go to this particular website, we're interested in here, this actually has a code of conduct for collecting insects and other invertebrates. This is actually published on the Royal Society of Entomology's webpage, but we've got the link here and you should read that so before you go out into the field you know what you're doing. Number one though, and I've not mentioned this before, whenever you go out collecting you should always get the landowner's permission. They may not be too chuffed if you start hacking through their vegetation collecting invertebrates.